The landscape of drug discovery is undergoing a profound transformation. Traditional methods, often reliant on two-dimensional cell cultures and animal models, frequently fall short in accurately predicting human physiological responses. This limitation contributes significantly to the high attrition rates in pharmaceutical development. However, a revolutionary paradigm is emerging. Genetically engineered mini-organs, known as organoids. These sophisticated three-dimensional structures derived from stem cells are poised to redefine custom drug testing, offering an unprecedented level of accuracy and personalization in our quest for effective therapies. For decades, drug development has grappled with a fundamental challenge, bridging the gap between preclinical findings and clinical outcomes. Two-dimensional cell cultures, while cost-effective, fail to replicate the complex cellular interactions, tissue architecture, and microenvironment crucial for accurate drug response. Animal models, though more physiologically relevant, often exhibit species-specific differences in metabolism and disease pathology, leading to unreliable translational results. This inherent disconnect necessitates innovative models that more faithfully recapitulate human biology. Organoids, with their ability to mimic the complexity of human tissues and organs, including cellular interactions and organ-specific functions, represent a critical leap forward. Their inherent three-dimensionality and cellular diversity provide a far more predictive platform for evaluating therapeutic compounds. The true power of organoids is unleashed through genetic engineering. By precisely manipulating their genetic makeup, scientists can create highly specific and relevant disease models, enabling targeted drug discovery. This capability allows for the introduction of disease-causing mutations, reporter genes for tracking cellular processes, or even gene knockouts to study gene function. The integration of advanced genetic tools, such as CRISPR-Cas9 with organoid technology, provides an unparalleled platform for dissecting disease mechanisms and identifying novel therapeutic targets with high fidelity. This precision engineering transforms organoids from mere tissue mimics into dynamic, controllable biological systems for rigorous scientific inquiry. Organoids can be genetically modified to model specific genetic mutations associated with a vast array of human diseases. This capability allows researchers to study disease, progression, and drug efficacy in a highly controlled and human-relevant environment. For instance, kidney organoids can be engineered to express mutations responsible for polycystic kidney disease, PKD, a common genetic disorder. Such models enable a detailed understanding of cyst formation, renal function deterioration, and the evaluation of potential therapeutic interventions directly on human-derived tissue. Beyond PKD, genetically engineered organoids are being developed for neurological disorders like Alzheimer's and Parkinson's, cystic fibrosis, and various cancers, providing invaluable insights into complex disease mechanisms. The concept of personalized medicine is rapidly moving from aspiration to reality, and patient-derived organoids, PDOs, are at its forefront. PDOs can be created from individuals with specific genetic conditions or tumors, enabling the creation of personalized systems that accurately represent the genetic variations within human populations. This allows for patient-specific drug evaluations, where the efficacy and toxicity of potential treatments can be assessed on a patient's own tissue in vitro. This approach promises to significantly improve treatment effectiveness by identifying the most suitable therapies for each patient, while simultaneously minimizing adverse side effects. The ability to test multiple drugs on a patient's own organoids before administering them clinically represents a paradigm shift in therapeutic decision-making. Organoids significantly enhance high-throughput screening, HTS, by replicating the complex architecture and physiological relevance of human tissues. This makes them ideal for evaluating large sets of therapeutic compounds with greater accuracy than traditional 2D cell lines. Their three-dimensional structure allows for more realistic drug penetration, metabolism, and cellular interactions, leading to more reliable screening results. Organoids can be used to test drugs for various diseases, including cancer, gastrointestinal disorders, and respiratory infections, providing a versatile platform for early-stage drug discovery. The scalability of organoid culture, combined with automated screening technologies, is accelerating the identification of promising drug candidates. One of the most critical applications of organoids is their ability to predict patient-specific drug responses with remarkable accuracy. By mimicking the biology of both healthy and diseased tissues, organoids allow for a comprehensive evaluation of compound efficacy and potential toxicity. This predictive power is invaluable in reducing drug attrition rates, as it enables the identification of potential failures much earlier in the drug development process. For example, cancer organoids derived from patient tumors can predict how a patient will respond to chemotherapy or targeted therapies, guiding clinical decisions and improving patient outcomes. This early identification of non-responders saves significant time and resources, streamlining the path to market for effective drugs. Genetic engineering of organoids provides an unparalleled tool for identifying novel drug targets and understanding their functional roles. By precisely manipulating genes within organoids, researchers can dissect complex signaling pathways and pinpoint critical molecular vulnerabilities. For instance, in the context of viral infections, genetically engineered organoids allow for identifying the role of host factors in cells closely resembling human epithelia, which are the viral point of entry. A notable example involves mutant clonal intestinal organoids, which have been generated to identify the cell surface protease TMPRST as a potential therapeutic target for coronaviruses. This level of mechanistic insight is crucial for developing highly specific and effective therapies. Drug resistance is a major hurdle in treating many diseases, particularly cancer. Organoids offer a powerful platform to study and predict mechanisms of drug resistance. For example, organoids harboring different RAS mutations can be used to test how these mutations affect the response to EGFR and MEK inhibitors, common cancer therapies. Studies have shown that organoids with oncogenic KRAS mutations displayed reduced sensitivity to these drugs, accurately reflecting clinical observations. This demonstrates the immense potential of using CRISPR-Cas9 edited organoid libraries for large-scale screens to identify resistance mechanisms and develop strategies to overcome them. This capability is vital for designing more durable and effective treatment regimens. Beyond efficacy, assessing potential drug-induced toxicity is paramount for patient safety. Organoids can predict adverse drug reactions with greater fidelity than traditional models, offering deeper insights into how substances interact within the body. Liver organoids, for example, are extensively used to study drug metabolism and hepatotoxicity, providing a more accurate assessment of how liver-targeting drugs work and their potential for causing liver damage. Similarly, kidney organoids can assess nephrotoxicity, and cardiac organoids can evaluate cardiotoxicity. This early and accurate toxicity screening helps de-risk drug candidates and prevents costly late-stage failures due to unforeseen side effects. Patient-derived organoids are increasingly being utilized as patient avatars in co-clinical testing. This innovative approach involves creating organoids from patients enrolled in clinical trials, allowing researchers to test experimental therapies on these in vitro models concurrently with the patient's treatment. This enables the stratification of patient populations and the personalization of treatment strategies even before large-scale clinical trials are completed. By enhancing the predictive value of preclinical studies, co-clinical testing significantly reduces the risk of trial failure and supports the rapid development of targeted therapies. It provides a powerful feedback loop, allowing for real-time adjustments to treatment protocols based on organoid responses. The transformative potential of organoids has spurred significant investment and innovation within the biotechnology sector. Companies like Hub Organoids are at the forefront, offering specialized services for drug screening, custom assay development, and clinical trials in a dish, using patient-derived organoids. These services span various therapeutic areas, including oncology, inflammatory diseases, genetic disorders, and toxicology, making organoid technology accessible to a broader scientific community. The growth of such commercial entities alongside robust academic research fosters a collaborative ecosystem essential for translating organoid science into tangible clinical benefits. Partnerships between academia, industry, and clinical centers are crucial for standardizing protocols, scaling production, and integrating organoid data into regulatory frameworks. As organoid technology advances, it brings forth important ethical considerations, particularly concerning the use of human stem cells and the creation of increasingly complex mini-organs. Responsible research practices, transparent consent processes, and ongoing public dialogue are essential to navigate these ethical landscapes. Looking ahead, the field is rapidly evolving towards multi-organoid systems, where different organoids are co-cultured to model inter-organ interactions, and the integration of organ-on-a-chip technology for dynamic fluid flow and mechanical stimulation. The synergy of organoids with artificial intelligence and machine learning for data analysis and predictive modeling promises to unlock even deeper insights into human biology and disease. These advancements will further refine drug discovery, making it more efficient, ethical, and ultimately more successful. Genetically engineered organoids represent a monumental leap forward in medical research and drug development. They offer an unprecedented level of biological relevance, enabling scientists to model diseases with greater accuracy, personalize treatments, and accelerate the discovery of life-saving therapies. By overcoming the limitations of traditional preclinical models, organoids are not just improving drug testing. They are fundamentally reshaping our approach to understanding human health and disease. This technology promises to deliver more effective, safer, and truly personalized medicines to patients worldwide. We urge all researchers, scientists, medical professionals, drug developers, and students to actively engage with this transformative field. Explore the latest research, consider integrating organoid technology into your studies, and collaborate across disciplines to harness its full potential. Your participation is vital in accelerating the translation of this groundbreaking science into tangible clinical benefits that will redefine patient care for generations to come. Hey, if you're enjoying this video and want to see more like it, don't forget to hit subscribe and turn on the notification bell so you never miss an update.